Hello everyone, my name is Dorianella, and today I'll be talking about the world collagen and the extracellular matrix. A little overview about collagen. Collagen is the main structural protein in the extracellular space in various connective tissue in the body. The specific protein is abundant in animals, vertebrates. It basically makes 25 to 30% of the whole body protein. It provides adhesive strength to the extracellular matrix, and it is secreted in several types of cells on the connective tissues, and the main one is fibroblast cells, and it can be found in different tissues such as tendon, bone, skin, intestinal tissues, muscle, cartilage, and embryonic skin. What makes up the structure? What structures are involved in collagen? And what makes it up? One will be the fibril, which makes up the bundle of the collagen. And then there's a triple helices, which is the collagen, the main collagen molecule. And there is the alpha chains, which is the long fibers, which also known as the collagen fibrils. And for different tissues, there's different types of structures, and those structures are the long fibrils, which can be made up of different types of structures. Can be it's 1, 2, 3, 4, 5, and there's also different ones such as the fibril associated with the interrupted triple helices. There's a fibril associated which forms bead filaments. There's the sheets, there's the anchoring fibrils, and then there's the transmembrane, and also there's there are basement membrane. For a basic structure of collagen, it is basically made up of a single alpha helix chain and 3 amino acids. Now, let's dive into the location in specific organisms and specific cells, and what is the physiological function and role of collagen. In animal, collagen is secreted in most connective tissue cells, which underlines the epithelium, and the tissue. Those tissue contains a variety of cells, and one specific cell that I put here is the fibroblast cell, which secretes and is abundant in the extracellular matrix. And that specific extracellular matrix that's around the fibroblast is composed of collagen fibrils. And the collagen polypeptide chains are synthesized on the membrane-bound ribosome and inserted into the lumen of the ER. And some physiological function of collagen is that it provides stiffness and elasticity for tissues such as skin. And the main key here is that it provides strength and structural integrity, which it helps organize the extracellular matrix. And some diseases that are associated with the malfunction of collagen have been genetic disorders, that the malfunction of collagen can cause genetic disorders, and some of them are the osteogenesis imperfecta, which causes weak bones, and causes irregular connective tissues. Also, there's the chondral dysplasia, which is a skeletal disorder, and the main one that's talked about in the book is the Ehlers-Danlos syndrome, which causes excessive looseness of the joints, and it damages blood cells. And also, there's the Alport syndrome, which is a basal lamina on an eye disorder, and it also causes kidney disorder. There's the Collagen syndrome, which is a mutation of a specific gene, and patient with this specific mutation have a protrusion of the brain tissue and degeneration of the retina. And some recent findings about collagen, the article that I found focused on the pathological gradings. Basically, they wanted to observe the collagen organization in renal cell carcinoma and see the difference in high and low and high prognosis tumors. And basically what this pathological grading is, it's a precise clinical need that's needed to identify prognostics and predict factors for patients with breast cancer or different types of tumors. And what they saw is that it's been known that the change in collagen organization in extracellular matrix has been linked to prognosis in breast, ovarian, and pancreatic cancer, but the organization has never been linked to the renal cell carcinoma. So they wanted to, they used a specific second harmonic generation-based imaging to measure the possible differences in collagen organization between high and low prognosis of renal cell carcinoma. And what they saw is that the collagen density and alignment was significantly higher in renal carcinoma prognosis 4 and renal versus renal prognosis 1. And that the biopsy sample using the SHG could match the classical tumor grading approaches. And it can also be clinically relevant for diagnosis. And also, the study basically will be required to investigate the functional role of collagen organization in renal cell carcinoma. And one of my questions is why is collagen pattern still being studied in renal cell carcinoma when collagen deposition or rearrangement has been linked to a non-renal cancer progression? Thank you.